So I'd like to thank everyone for their time today. And what I'm going to do is give an overview of a bit of the research that we have going on in my lab. Uh, okay, so just to give everyone a little bit of information of what we do in my lab. So I run a cellular biomechanics lab, and the overall goal here, as I have, is to link the body or link mechanics to the body at the cellular level. And we've been focusing on the human body, but I will say that some mentions that we have are actually going beyond the human body and actually looking at non-human systems at some point. So just as an FYI, those are some collaborations that we're looking for here at some point here. But I joined the MAE department in 2015. And again, the overall goal here is just to elucidate the physical mechanisms that really drive human physiology and pathology at the cellular level. So in our lab, what we do is we combine mechanics and cell biology and molecular biology, and we really want to learn the implications that mechanics has, again, for the physiology and pathology. For some active research projects that we have currently going on. So we've currently been we look a lot with endothelial cells, and just as a FYI, endothelial cells, those are going to be the cells that line the inside of all of your blood vessels. So any blood vessel that you have in your body is going to be lined with endothelial cells, okay? And being that they do line the inside of blood vessels, they're constantly exposed to fluid shear, okay? So we do a lot of device design. We have a lot of interest in looking at the influence of various fluid flow regimes on endothelial cells, laminar, turbulent shear. There's a regime called disturbed flow as well, which I'll talk about briefly in one of my projects here, okay? Transendothelial migration is the process of white blood cells actually crossing over the vasculature. So that's important for your immune response, right? Inflammation, right? Anytime you get a paper cut or any cut, white blood cells will actually have to move from the circulation, cross the vasculature to try to heal the wound or remove any debris or bacteria or potentially harmful invading pathogens here. The blood-brain barrier is another one, diabetes, and a recent one that we started, maybe a couple of years ago, was the gut epithelium. And this is actually in collaboration with one of our collaborators at the Mayo Clinic, actually in Minnesota, in Rochester here. And on the side here, that handsome person on the right is me, and I just have a few little representative images here of what we get from our lab. Now for teaching, based off of this theme of really linking the mechanics and biology, okay, overall, this is what drives me. And these are things that I think about. Some classes that I've taught thus far, so I've taught continuum mechanics, biofluid mechanics, which is essentially a link of fluid mechanics, and we look at fluid mechanics of the vasculature of the heart, various other organs such as the kidney. Last semester, actually, we opened a special topics course, continuum biomechanics. Okay, and that was essentially a continuum mechanics class, and we looked at mechanics more applied to the body, and mechanics of biostructures too, which I also taught is more of a cellular mechanics class here. And the way that class was structured, essentially what we did was we started off with more of just a pure cell biology concept, and then we applied mechanics to cell biology as well there. And all of these classes, with the exception of continuum mechanics, these were all classes that were designed and constructed on my own. So these are all new classes that were designed and built to contribute to the current BME curriculum that we have in the Mechanical and Aerospace Engineering department here. Now, what I want to first do is just quickly go over one of the main methods that we use in the lab and some of the metrics that we use to really characterize our cells here. So what you'll hear throughout this presentation are attractions and intercellular stresses, okay? What are tractions? We use the method traction force microscopy to measure tractions, okay? These are the forces between the cell and the substrate, okay? Intercellular stresses are going to be the stresses between the cells themselves. We use monolayer stress microscopy for that, okay? And we can calculate the normal and shear intercellular stress. I like to think of tractions and intercellular stresses interacting with each other through a tug of war here, okay? Now, what I'll show next, keeping that in mind. So one thing I want you all to hold on to, just remember tractions are between the cell and the substrate. Intercellular stresses are going to be between the cells themselves. So we have cells that are joined together by cell-cell junctions. They actually transmit stresses through their cell-cell junctions here. Now, what I'll show next are a few movies that I have here. And as you'll see, we have a control and a fluid shear. So control, we can understand that are just cells in static conditions. So essentially these cells are on a surface. There's no external mechanical force. Now you'll see these little black circles or black shapes. These are the actual cells themselves. The colors that you see are going to be the normal intercellular stresses superimposed on those cells. So these are two images imposed on each other. And what we'll see are just how the intercellular stresses change in static conditions here. So as this is planned, the main point of that, the intercellular stresses certainly fluctuate in space and time, but also what I want to get across is that for any cell type, as long as it's an adherent cell type, we can generate a mechanical phenotype if one exists. And now we'll look at fluid shear. And on the right here, these were just cells exposed to a laminar fluid flow. Okay, and the main point is that as you can see, there is certainly a drastic reduction in the intercellular stresses here. Now to give more of a flavor of what we can get from our method as well, what I'm showing here next are some histograms of the orientation of the cell bodies and the intercellular stresses. Quickly go through that. So we have our control, have our fluid shear. The main point here is that under control or static conditions, there's no preferred orientation of the intercellular stresses in cells. Under the fluid shear, there's an alignment, but what ends up coming out is that we found that the intercellular stresses end up aligning along the direction of fluid shear before the cells themselves. Now, this is another project here looking at the stirred flow. And what we did was we developed what's called a step flow chamber. This is commonly used in the field, and essentially you literally have a step in the chamber, and it's, it's interesting and it's useful because we can actually look at multiple flow regimes in one experimental setting here. So not to go into a lot of detail, the main point is that, as you can see, I have the zones 1, 2, and 3. Zones 1 and 2 would be what the cells would experience and what we call the bifurcations or branching regions of the vasculature. These are going to be regions where a lot of pathologies occur. Zone 3 are going to be the type of fluid shear stresses that cells experience in your straight parts of the vessels. And these are the regions less likely for various pathologies to occur here. So the main point is that in the regions where you have more complex flow regimes, you have higher intercellular stresses. Regions where the flow is more steady, you have lower intercellular stresses here. What I'm showing next are just some still images here. This is from our Organoid Project, our collaboration with the Mayo Clinic. And essentially what we've been able to do, and just to backtrack, organoids are 3D tissues, okay? And what we've been able to do is take these 3D tissues from the mouse intestine and planarize them and measure the tractions here. And what I have on the left are just these organoids that have been planarized, spreading out on various substrates. And on the right here, we actually measured these tractions. And we're excited about this because no one has actually measured the tractions with these planarized organoids before. So that's something, a breakthrough that we're really excited about. Now for the short term and long term here, what can we do in our lab? We can generate a mechanical phenotype as long as the cell type is anchorage dependent here. although I didn't show any of it in this presentation, we do device design. We have a 3D printer. I mentioned earlier, my interest is really looking at various fluid flow regimes on endothelial cells, but it doesn't have to be fluid flow, it can be stretched. We've also looked at fluid shear and stretch, so we couple modes as well here. As far as short-term and long-term goals, certainly we're looking at, looking for more medically related collaborations, but we're open-minded here. Maintaining our focus on the vasculature, especially as it relates to COVID-19. So there's a lot of literature out that shows that some of the long-term effects of this virus is that it actually damages the vasculature. And, some people who have actually succumbed to this virus have, hypercoagulation, or their blood clots that essentially form in region of the vasculature all throughout the body here, okay? And as I mentioned earlier, I certainly envision my lab moving beyond just human cells, but moving more towards non-human biological system, plants, animals, whatever it may be here. For my acknowledgements, what I presented was funded by the NIH and UCF startup funds. And of course, I'd like to thank my lab for their help. And I'd like to thank everyone for their attention. And that's it.